My biology role model is Dr. Yoke-Fun. Dr. Chan's passion for science deepened during her undergraduate years at the University of Malaya in Malaysia. She credits her involvement in developing a diagnostic test for thalassemia, a blood disorder that affects the body's ability to produce sufficient hemoglobin, with truly igniting her interest in research. This experience helped her realize that her love for scientific exploration. Her specific interest in virology was later shaped by a fascination with historic pandemics such as the Black Plague and the Spanish flu. The emergence of new viruses has further fueled her dedication to continue research in this field. During her undergraduate studies, Dr. Chan majored in biomedical sciences at the University of Malaya, Malaysia. She then pursued her PhD in the Department of Medical Microbiology, Faculty of Medicine, also at the University of Malaya. Some of the toughest challenges Dr. Chan encountered involved securing consistent funding for her research. Local funding was often limited to 1 or 2 years, making it difficult to plan long-term projects. Another significant challenge was the loss of many talented students from her lab, as they chose not to pursue postgraduate studies, depriving her of promising researchers. Additionally, while Malaysia may have the necessary technology for advanced research, there is a lack of expertise, further grounded by senior researchers being unwilling to share their knowledge and collaborate. Outside of her work as a scientist, she likes to exercise and watch movies with her family. She also enjoys traveling with her family and exploring a variety of different dishes around Malaysia. I chose Dr. Chan because she's an inspiration for women in science, particularly Southeast Asian women. As a Southeast Asian woman myself, I find her story incredibly motivating. Her work as a virologist and in vaccine development is not only fascinating, but also represents an intricate field that I've always wanted to explore. Another reason I chose her is her commitment to academic outreach, as she actively contributes to educating the next generation of scientists and medical professionals. Beyond her scientific accomplishments, I admire her ability to balance personal and professional life, taking on diverse roles as a scientist, educator, and mother. She defies the stigma that women in STEM must choose between careers and family, proving that they can thrive in both realms. Dr. Chan is currently working in microbiology as a virologist. She's focusing on the enterovirus A71, which causes hand, foot, and mouth disease. Additionally, she's conducting further research on brain infections. The main purpose of this article was to assess the stability of various enteroviruses linked to hand, foot, and mouth disease on different types of children's toys commonly found in kindergartens. The figure on the right shows the results for enterovirus A71. The virus strains were first cultured in human rhabdomyosarcoma cells, a type of sarcoma, using Dolbeco's modified eagle medium, supplemented with components like penicillin, sodium pyruvate, and streptomycin. The cells were then freeze-thawed twice, centrifuged, and viral titers were determined by measuring the median tissue culture infectious dose, calculated using a Reed-Munch method, a technique that's used to calculate the concentration of a test substance that produces an effect in half of the test units. Commonly found kindergarten toys were then taken and sorted into their various porosities. These materials were then cut into 2 by 2 centimeter squares and disinfected using 70% ethanol. 30 microliters of a solution containing the virus was pipetted onto the toy surface, allowed to dry for an hour, and then stored at 4 degrees, 24 degrees, and 35 degrees Celsius. Relative humidity was kept constant, and the samples were collected in triplicate from each surface at 0, 24, 48, and 72, and 96 hours. An atomic force microscope was also used to image the toy's surfaces. The stability of EVA71 was inferred through half-life calculations, and the figure shows that EVA71 was most stable at 4 degrees Celsius. The experiment concluded that factors such as porosity, temperature, and environment affected the stability of viruses. The overall result shown in the figure represents how EVA71 was most stable at 4 degrees Celsius. Some of the main things Dr. Chan has uncovered so far are that Dr. Chan's research has also revealed that heparan sulfate acts as an attachment receptor for EVA71, a discovery that could lead to potential treatments for hand, foot, and mouth disease. Our future research will continue focusing on EVA71 to find a cure for hand, foot, and mouth disease and help accelerate the diagnosis of neuroinfections in Malaysia.